Fundamentals of EEG biomedical signal processing and applications. Here we will discuss about different types of modalities of the brain signal with more focus on EEG signal and other biomedical signals as well, and how these signals are processed and applied in different applications. That will be the agenda of this lecture. So in a title slide, as usual, you always find a picture, and that picture, the first one, just to sum up that these are the modalities which we get. We have been studying for last 7 long lectures. EEG, look at here at the scalp. ECOG is a cardiogram that is little deeper, and then implant local field potential. I hope you have been attending all these lectures. So LFP is the local field potential, and you get a better and best quality of information in the implant local field potential, where you insert some microarrays, microelectrode arrays in the form of integrated circuit inside the brain, and you try to collect a very localized information. So the process, the other stages which have been highlighted in this slide, the title slide, are all the general purpose steps. There are a number of subjects. Subjects may be related to Alzheimer's disease sample, a mild cognitive impairment sample, or healthy control sample. So you may have a number of subjects of some of those are suffering with some disorders, impairment. Some of them have some disease, brain related disease, and some of them are healthy samples. So we collect the data, we pre-process the data. You know it very well now. Why do we pre-process? We do not need to repeat again. After pre-processing, we extract the suitable features using a feature extraction methods. Once we extract the features, those features may be based on Fourier related features or wavelet analysis features. So we can apply frequency domain analysis, what is called as a frequency analysis. We can also apply wavelet domain analysis, which also provides time frequency analysis both in the wavelet domain, and any of such analysis that will produce the features will be subjected to the classifier. So with this generalized block diagram of brain signal processing, let's begin with recapitulating about EEG. We all know that electrical potentials which are expressed in microvolts, we can extract those potentials from scalp outside the surface. Generally, we use 1020 system for recording the EEG, which has usually 19 electrodes. But there are other systems, they have more than 19 and up to 61 active electrodes in the surface area. This diagram you have seen a number of times in previous lectures. I have already described what these F, P, C, T, O, A denote. F for frontal, P for parietal, C for central, T for temporal, O for occipital, and A for auricle. So these are the the letters which you can make out that what are the electrodes or the Montage's name. And odd number electrodes are generally on the left side. Look at here, and even numbered electrodes are generally on the right side. Zero is always along the middle one, that is on the central one. So this is the general standard, and there are systems where you have more than like 61 electrodes in this particular case, but the nomenclature here is also the same nomenclature. EEG as I told you that in this lecture, we'll be talking about all those modalities, their processing, but some of those will be untouched. Some of the aspects which we have not studied will be covered. Like EEG is signal which is not deterministic. I hope both of you and all of you know the difference between deterministic, random signal, stochastic signal. So this is not deterministic. You cannot determine, the amplitude and frequency after certain interval, what amplitude and frequency it will have. EEG signals are therefore stochastic. They are assumed to be linear in many of the applications. For simplicity, through this is also not a always correct. They may not be always linear. So since we analyze the signal in discrete domain, you know very well that what do we mean by analog, discrete and digital. So generally in the discrete domain, the linear system is expressed as yn is xn plus xi minus 1. Yn is the response, xn is the input, xn minus 1 is the value of previous input or the value of, yeah, just past input. And here you look at here to the power 3. So wherever there is, the exponent is 2 or more than 2, it no longer remains linear. It becomes non-linear. So there are different types of systems. When we mention about EEG signals, they generally cover the following spectral range, frequency range, and that also has been already covered. What is new thing here is each spectral is associated with one of the example, like delta. Delta was also explained to you that during meditation or when we are in deep sleep, delta is generated. Delta generated. And what is the range of delta? Zero point 0.1 to 3 hertz, very small frequency. And probably you are aware that the amplitude is higher. Then comes theta. The range is 4 to 7 hertz. And it is generally expressed or emitted during the sleep. But when there is a deep sleep or when you try to focus something, when you are trying to concentrate something and meditating, in that case, delta is generated. Alpha wave, it ranges from 18 to 13. And during restful, when you are not doing anything, but of course you are not sleeping, relaxing state, it decreases when eyes are open. Remember, so when you are in relaxed state, when you are trying to rest, but when your eyes are open, then the waves are decreased. But when your eyes are closed, you can clearly see the presence of alpha waves. Beta, it starts from 14 to 19. Look at here, delta, theta, alpha and beta. So alpha, beta, gamma are in sequence, but before that delta and theta comes. So during the restful, you get alpha, and beta, which starts from 14 hertz, goes till 29 hertz. This is found during some mental activity, like you are doing some mathematics, computer based mathematics. There the beta is generated. Talking about gamma, more than 30 and up to 50 hertz, you understand if it goes to 50 hertz, there may be some interference by the power line signal. I have already explained in my previous lecture. So in any higher order brain activity, when you involve some critical thinking kind of thing, also a triggering mechanism alerts the brain to recognize a picture or a sound. So some higher level mental activity is going on. That time gamma is generated. And then EEG, which is less than 0.1 hertz, which is written here, it starts from 0.1 hertz, the below which generally baseline wander noise. So the signals which are below zero point 0.1 hertz are considered as the noise, not wanted signal. Let's talk about evoked potential, what we denote by ERP. We also call it as an event related potential. If you can recall, we have done 2 lectures on P300 BCI. And in P300 BCI, we use semi invasive modality, especially the evoked potential and event related potential. And that is actually EEG in response to some stimulus like visual, audio, etc. So ERP may not be always generated by the semi-invasive. We are still talking about purely non-invasive. Where EEG is there, wherever we talk about EEG, that is always non-invasive. So that how do we get this event-related potential? You all know it very well to some stimulus like visual audio impact. The EP, that is evoked potential, is evoked with the brain is still active doing other things. It means the brain can be performing some other task also along with responding to the stimulus. Remember, so sometimes when brain is engaged in some critical thinking or computational or some mathematics, then it is engaged in that particular work. But when responding to stimulus, the brain can also be involved in some active task. Therefore, we need to use ensemble averaging because there may be other task or the activities where brain is engaged. So what do? We use ensemble averaging, median filtering, or principal component analysis and similar methods to reduce the ongoing EEG. Remember, because if we are interested for recording only evoked potential, we are not interested for other signals. What do we mean by other signals? Because when the brain is responding to the stimulus generating evoked potential, at the same time, it is also generating other responses to the different mental activities. And therefore, we need to reduce those ongoing EEG responses. Otherwise, will not get the pure one. This diagram is very well known to you, like stimulus is there. This is P1, N1, you know about this, P2, N1, P2 amplitude between N1 and P2 peak, and then P3 we get what we call as a P3 latency. So this all we have covered about what do you mean by latency, what do you mean by P1. So EPs are, that is evoked potentials are normally analyzed in time domain. There are a number of peaks in evoked potential after reducing ongoing EEG. So whatever analysis we make over the EP evoked potential, we generally start performing after reducing the ongoing EEG activities. And we can do it through averaging or ensemble averaging method. And then finally, what we get, the peaks are identified as P1, P2, P3, N1, N2, N3, and so on. Sometimes the peaks are identified using the latency. You can say P300 and 200. And it is known that the third positive has latency about 300 millisecond. And the corresponding BCI is called as P300 BCI. Each peak, whenever you get a peak in evoked potential, will always have latency and the amplitude. Certain latency after how much time after the stimulus was generated, the brain has responded. That is called a stimulus latency, and of course it will have certain amplitude. Amplitude is the amplitude of the peak in microvolts. The what are the different types of evoked potential? Evoked potential can be of small amplitude, lower amplitude, and lower latency, smaller latency. Latency is small. Evoked when the stimulus is brief auditory stimuli. There may be other type of evoked potential where you generate some small auditory stimuli, sound, maybe clicks or narrow tone burst. You can provide the user or a subject a narrow stimuli, narrow click or a narrow tone burst. You can click a mouse, you can just press a button, that kind of clicking sound can be given as the stimuli. In third case, it consists of about 6 waves in the first 10 millisecond after the sound. The frequency range is about 100 to 2000 hertz. So these are the things you see 1, 2, 3, 4, 5. This is how you get the peaks. This dB is nothing but expressed. The strength is expressed. The amplitude is expressed in the dB, and then this side is the timing, that after how much time you are getting the peak. So that is your latency. There comes another name called as somatosensory EP, evoked potential. What is this? This is related to tactile stimulation. You understand very well what do we mean by tactile stimulation. When we touch something, that touch kind of stimulation is given to the subject or the user. That is called as tactile stimulation. How can you give tactile stimulation to the subject? By electrical stimulation of median nerve at the wrist or nerves at leg. Some point at the wrist, you can just give some touch, tactile stimulation to the subject, or somewhere at the in the nerves at the leg, you can give the sensation, touch sensation. The total duration is generally 400 millisecond, but only that first 20 millisecond part is more important. The response, what we get from the different points after providing tactile stimulation, are all localized on the scalp or the hand area of sensory motor cortex, contralateral to the stimulated wrist. The EP spectral component, rather content, are from 100 to 2000 hertz. After talking about what different types of evoked potential are, let us talk about what are the different features. The features are computed from the signal. Once we get the raw signal might not be very useful. So we recover and we pre-process the signal. We discard some unwanted signal, and then features are extracted. What is the significance of feature? Features always represent the content of the signal, and therefore that can be reproducible. Reproducible means what? If we have extracted the features from an EEG signal, by combining those features, we must be able to generate the same signal. So it is reproducible. Smaller dimension than the signal, the feature dimension will be lesser than the signal dimension. Otherwise, you can directly work on the signal dimension. So we'll have less variability, less content when we directly work on the signal, and the dimension will be also more. Interclass variance is high and intra-class variance is low. As far as the features are concerned, we have to consider that kind of feature where we have such things. The evoked, this is one interesting diagram where you can see the, it highlights all the stages with examples of some different stages, like evoked experiment where subjects are there. They watch the stimulus. This is included in the diagram. Stimulus causes the persons to react, and the reaction is recorded as waves. So EEG is acquired here with the help of electrodes. Sent to the pre-processing, we remove the artifact and we improve the signal quality and improve the SNR. We extract the features. How do we extract? The features may be time domain features, frequency domain features, or time frequency domain features. Like frequency domain features are obtained by using the Fourier transform, FFT. Time frequency domain features can be obtained by applying wavelet transform. Then we send to the classifier where we can predict in a different manner that the features are representing positive, negative, and neutral. The meaning is, if there is some disability or disorder that is indicated will be, or if there is any possibility of brain tumor cancer or some abnormality inside the brain, will be indicated by positive. If there is nothing like that, it will be negative. And if it's a healthy person, then it will be neutral. So what are the machine learning methods which are generally used? Variety of ML techniques are used for EEG signal processing and feature extraction. SVM, support vector machine, KNN, K nearest neighbor, naive Bayes, random forest method, extreme learning machine, ELM, and then currently we are talking about deep learning models, because whenever there is a dimensionality increase, we always use the deep learning model. because especially with the EEG signal processing, you have a huge dimension involved, and therefore the huge dimensionality problem can only be addressed by the models like deep learning. So what are the different examples under DNN? CNN called as convolution neural network, recurrent neural network, RCN, deep belief network called as DBF, DBN, artificial neural network, and long and short term memory called as LSTM. So LSTM is actually very similar to RCN, where you improve something and it becomes LSTM. What are the different frequencies? Some examples are given here. Like, as I mentioned, that what are the different types of features? Time domain, frequency domain, and time frequency. So under time, like statistical parameters will be there, like mean value, standard deviation, average value. Those values can directly be computed in time domain. There is one more parameter called as Jorth parameter that is also calculated in time domain. In time frequency, ST Fourier transform, short time Fourier transform called as STFT, Webelet transform, Hilbert Huang transform. So these are the examples where time frequency features are extracted. In the pure frequency domain, power spectral density, PSD, differential entropy, higher order spectrum, higher order crossover, where in many of the literature you will find power spectral density as a common feature used in the application. So a robust and enhanced representation of signal is a very important thing when we extract the features. Means when we extract the features, the representation of signal by using those features must be robust. There must be some general purpose, definition of signal by using those features. And what is more important regarding robustness is invariant to changes in noise. So when we have extracted the features, even if there is a noise, we are able to extract. So the signal is invariant to changes in noise. Even if we scale up or scale down the dimension of the signal, so it is independent of the scaling factor. It is independent of change in noise content. Then we'll call robust set of features. And the moment when we get robust set of features, the performance of the method becomes automatically towards robust. So sample features are like mean, which is not very useful, generally set as 0. Standard deviation, energy, which can be computed by using variance after setting mean to 0. Correlation of EEG signal, sometimes we use correlation. Correlation is very important a feature in communication system as well. Correlation of EEG signal with the template EEG signal, that is sometimes also used as features of EEG signal. Autoregressive coefficients can be determined by using linear feature extraction method. AR coefficient called as autoregressive coefficients that can be used for biological signals known as parametric method, because method is based on suitable parameters which are used in this linear feature extraction method. A real valued zero mean is always zero, stationary, non-deterministic, autoregressive process of P. Suppose there is a signal, rather there is a feature which is the real value. Its value is real. Mean is zero, stationary, non-deterministic, of course stochastic, an autoregressive process. If you can go back, then you can get your originality in terms of signal. So this is defined in discrete domain. K equals 1 summation of 1 to p ak x n minus k plus en, where p is the model order. This p is the order of the model. You may have second order model, third order model. It is very similar to the filter design which we use in the analog domain for pre-processing and other operations. Xn is the data of the signal at sampled point. Xn original one. Here xn minus k is a delayed one, and ak here it is actually ak. K is the lower script, are the real valued autoregressive coefficients. Why it is autoregressive? Why it is called autoregressive? Look at xn minus k. So when you put k equals 1, it becomes xn minus 1. When you substitute k equals 2, it becomes xn minus 2. When you substitute k equals 3, it becomes xn minus 3. So it is calling the previous or the past values. So therefore you call it as autoregressive coefficients. That is of this. And En represents the white noise error independent of past samples. En is contributed as noise added with all those samples. Remember, there is only one sample. It is actually when you put k equals 0, it becomes xn. But since we are involving other past values also, because of we are using autoregressive model, and therefore this function is coming like this. Otherwise, it will only be, xn. And then power spectral density values are also used as features for EEG signal. I have already told you power spectral density that is in frequency domain. Different methods are used in order to obtain or determine power spectral density like periodogram, Welch and AR based method, Welch method, autoregressive based models are there by which we calculate the power spectral density. There is no more no, there is more spectral power in lower frequency during maths activity as compared to the resting. There is more spectral power in lower frequency during maths activity. So when the brain is engaged in the some mathematical related activities as compared to resting potential or resting activity, there will be more content in the low frequency side because both of the signals or the signal in both the activities, either resting or mental activity, is always of low frequency, but more spectral power can be seen because in mathematical activity, that energy which is spent is more. So this is one example for, I think if I go back, this resting EEG and the second one is maths EEG. This one, first one is your resting one, second one is maths EEG. So you can clearly see here that after certain interval where you get a significant peak, you know, you can see the, the difference here. You look at here that it is going slightly till this one, but it is even crossing 30. It is, if you compare this to somewhere in the, especially in the lower frequency, we are saying lower frequency side, look at it 20. So 20 is here very near, but here 20, before 20, lot of signals are there. Even if you compare before 10 and before 10, you compare this and this one, you will find more spectral power will be, there before, I mean, during the low frequency. This is because of, in this case, the mental activity is mathematics, whereas in this particular case, the activity is resting activity. There is one more term which we use as the analysis of biomedical signal called as peak frequency, where when we say peak frequency, there are two terms automatically covered. One, frequency, second is the amplitude. The actual frequency value of the peak, and what is amplitude? Amplitude value of the peak. What is the amplitude of the PSD? If you go here, go back to the previous one, look at the peak. So just measure the amplitude value, that is your peak amplitude, and then frequency. Width, width of peak computed using some threshold like dB. So if you see here, this is the width of the lobe, which you are getting as a signal that is always expressed in dB. We also use the area under the peak. We can calculate area computed as the sum of PSD values from say plus minus 5 hertz. For example, like this. This is if you are interested from at zero point 0.5 hertz, then plus minus zero point 0.5 hertz at that frequency you can calculate. This is what is talked about. Look at here that this is one peak and this is also peak. So you can measure different frequencies at which peaks are occurring. You can measure the width also, and all these parameters are used in the analysis of feature. Another parameter which is coming as asymmetric ratio PSD. After obtaining PSD, we can obtain the asymmetric ratio. ASPSD, that is calculated as P1 minus P2 divided by P1 plus P2. where what is P1? PSD of one channel. P2 is the power in another channel, but in opposite hemisphere. So, you can just, if you take on front side, just go on back side and take from both and get this value as the asymmetry, because when you are on both are on opposite side, there will be asymmetry. So sometimes we calculate the asymmetry ratio, how much they are asymmetrical, how much they are not symmetrical. That is indicated by, look at here, if this is one side, the other side will be this one. So useful, this particular ratio is also useful, considered as a useful feature when the mental activities demonstrate hemispheric difference. When you have, that in a hemisphere there are different lobes. So between 1 and 2, if it reports a difference, that will be highlighted by the asymmetric ratio. If it is higher means the inter-hemisphere difference will be more. So this is indicated by the mental activities and different kinds of mental activities between in different areas. Maths activity exhibits a higher power spectrum in the left hemisphere generally, whereas visual tasks do so in the right hemisphere. Hope you understand the reason for this. The visual tasks do so in the right hemisphere. Why? If you have seen the brain diagram, so occipital lobe is this side. So in the occipital lobe, vision related task is performed this side. And therefore this hemisphere visual tasks do so in the right, whereas in the left one, this side, maths activity are performed this particular area where whatever is the person that is more responsible for applying such or engaging certain activities. ASPSD using O1 and O2. I mean, there are two channels like O1 and O2 means there are two electrode positions will be higher for maths activity as compared to visual activity. Because if you go for visual activity, it will be till here. So the difference between that will be higher. This is one example like a PSD of O1. What is O1 is highlighted there, occipital area. O1 means occipital area here, and then PSD of O2 and PSD of O1 after rotation, because if you can rotate the portion, then you can calculate, and then APSD for subject maths and take the average as 0.32, and for the rotation activity sum is 0.143. So one is for the mathematics activity, and other is for the other one. Spectral correlation. Other feature is spectral correlation or coherence. This diagram is indicating how do we evaluate the spectral coherence, that is the correlation between the two signal, how do they match, and how do we evaluate this side one signal, this side other signal, and between frequency and magnitude domain, how do we see their coherence, correlation, how they match with themselves. So it's a standard tool measuring spectral coherence in the signal processing. So for EEG also, the correlation studied with how can we correlate it? The correlation can be computed with the help of power spectral density values. Like in a simple MATLAB kind of program, X and Y are the PSD of two signals. One is for rotation, other is for maths activity. You can simply write a command like R is equal to correlation coefficient X and Y. That will give us all the correlation coefficients and will indicate how much correlation is there between one signal and then signal which is rotated. So the spectral correlation of object rotation in channel O1 and O2. So SM or O1 is the correlation between these two PSD is 0.95, and between 2 and then other area SMR O2 is 0.94. So this is one metric by which you can calculate the correlation between the signal after rotating. Other one is joint time frequency analysis. There are three approaches under this. The first method, which comes under joint time frequency, is linear non-parametric methods, which is obtained by linear filtering such as STFT and wavelet transform. You know that time frequency. It is obviously wavelet transform always falls under this. Quadratic non-parametric method, quadratic and non-parametric method. Improved time frequency resolution is obtained in this method such as Wigner-Ville Wigner-Wille distribution, and other similar techniques. Other joint time frequency methods are parametric methods. Assuming that the observed signal can be obtained from time-varying statistical models such as autoregressive model. We have briefly talked about autoregressive model where you use autoregressive functions like x and -1, -2, -3 to evaluate how and to consider what were the past values of the signal at certain sample. So this is, one example of where the signal in part A is EEG and signal in part B is time frequency analysis. If time is also there, frequency is also there. So this might also be wavelet transform of the given signal, and that gives a lot of coefficients. These all square boxes which you can see are representing the wavelet coefficients or coefficients of the transform, which is applied in time frequency or joint time frequency domain. So let's talk about a task where a psychologist want to measure patient's anxiety. The person is having anxiety, and the psychologically one expert wants to evaluate, assess how much anxiety the subject has. So it will, the study will be based on measuring the brain activities associated with anxiety. So you know that in which part of your brain anxiety is associated, and that corresponding wave which is actually used in the study of anxiety is alpha wave. So alpha brain waves are generally the good measure for assessment or study of anxiety, and the subject, that is a person, will respond to audio or visual cues. He will be given some audio or visual cues, some pictures he will be shown, and he will be responding after watching and seeing the hearing, listening the audio or watching the picture. And accordingly, based on the response analysis, feature extraction, classification, and analysis, one can evaluate that how much a person is undergoing through, anxiety situation. We can take psychophysiological readings such as ECG and EEG both with the subject whenever the subject is performing the task. You are giving a task to the subject that watch this picture or hear this sound and respond. So you may record either EEG as well as ECG, and you can find out that how much anxiety a person is, having. ECG, you know that generally the persons who are having cardiac problems, they are advised not to worry, not to have stress, not to have anxiety. So whenever there is anxiety, stress or tension, then it is also reflected in the ECG. Similarly, EEG also reflects what is going on inside the brain when the person is suffering with anxiety disorder. Readings which are recorded from the person's brain are manipulated so that we can generate a data by using a statistical analysis, by using suitable statistical parameters, and we can come to certain conclusion. So this is one example where this first one is the normal EEG of left hemisphere, left side, and right hemisphere EEG is this. And when you compare with the person suffering with depression, having certain anxiety, if you compare with the left one, and right can be compared with right one. So what we can observe is looking very similar, though even in all the signals some artifact is present. But when actually compare carefully, you compare that at 400 C here, and even at 600 when you can compare, and maybe at 1400 also you can compare, the peaks are different. So there is a clear cut difference which is visible when the person is normal and when person is having some depression. Similarly, in the right hemisphere also one can compare that at different points there the difference can be clearly seen. For example, you see this window from 200 to 600, but here your window is expanding from 200 to rather 800, and the signal is very different from what is there in the normal right hemisphere area. So you can just give a task, perform this task, and you say that watch this picture carefully, interpret it, or listen to the sound and interpret it. So then you can evaluate whether the person does it with the patience, does it with calmness, or just struggles because of he undergoes with the anxiety and depression. So that can be very well studied with the help of biomedical signal processing. This diagram is a popular diagram, well known to you, that when we deploy a number of electrodes, and you can see that these two, this side one and that side one, these are nothing but reference electrodes. You are aware of, we use the reference electrodes at different points. So the measurements which we do, because when we want to study the anxiety level, how much anxiety is there, we have to do some measurement. So what will the measurement include? It will be EEG for alpha wave brain activity and ECG for stress level, especially when we are interested to determine the anxiety on the basis of brain activities. How does anxiety change? How does anxiety, affect the different parts of the brain? So alpha wave is recorded. And when you want actually to understand stress level, so one can even study ECG signal. EEG is obtained using a biosemi in an experiment we are talking about. This particular paper has talked about some work where they studied on the anxiety, where a biosemi device, which is a 128 channel active electrode device, was used, and the system recorded the data with active view program. And E prime was an interface that presents a series of visual and audible cues to the patient. So you can say this is a stimulator, E prime, and that presents a number of visual and auditory cues, the signal or a stimulator to the participant. So in synchronous to these cues, along with the cues which are presented by E prime, triggers are also sent to biosemi EEG. Some triggers from external sources are also given. Triggers are set out to the printer port using its 8-bit channels, and these may appear as binary labels or a number. Trigger information contains record oblique pass command and task identification. So what is this biosemi active view? Because that is used by this work performing a task to evaluate the anxiety. The biosemi active electrodes, each of them have a tiny electronic circuit, a very small electronic circuit. It has also ability to eliminate 50 Hertz interference picked up by the electrode wires, kind of a power line interference. 128 channels give detailed coverage. How many channels are there in biosemi? There are 128 channels. So because the number of channels is huge, so the coverage is very good from all parts of the brain. You can get the signal. Obviously, you can understand in the second stage, a step that when you have more coverage, huge amount of dimension will always be there. So 8 channels for auxiliary electrodes, left and right eye blinks are also considered. This is what is biosemi, and you see here it has some electronic circuit also, so that it can pick up and it can record the signal by providing also some kind of signal processing, say, some kind of to some level amplification and other basic pre-processing. This is the plot which was generated by what is called as active view. Active view displays your picture like this, that how many samples are there at different, uh, at different values, different frequency. You can select the channel and then you can watch. You can see that keyboard replace eyes open when you are performing certain with the keyboard, when you are reading, when your eyes are open, when your eyes are closed. So all these are different plots for the different things. This is very similar to the previous one. And then comes data analysis in a particular task we are talking about presented in this work. So data analysis was done in this work by using a software called as BESA. And this BESA stands for Brain Electrical Source Analysis. And you look at there that it will tell you the brain's position, what is the conductivity of brain, how much thickness is there, what model of method is being used, what kind of analyses, what are the subjects, what are the different values, or how do they what are the signals which are coming from different electrodes and montages. They are all highlighted, and they'll give some analyses as well for the modalities. These are all the the channels or montages from which the signals are coming. This is some more without the artifact using a 50 Hertz notch filter and fast Fourier transform on all the channels when eyes were closed. So the uh, you know paroxysmal non-elliptical epileptic spike activity at T3. You know about T3 left temporal lobe because odd number goes in the left side related to emotional explosive behavior and memory problem. So if someone has this kind of problem, it will be indicated by the spike at where T3. So whenever you get certain kind of spike at T3, that indicates the presence of some emotional explosion in the behavior or having certain problem in the memory. Can also look activity. We can also look at the activity generated in different areas of brain. What are the different you see here from different areas of brain being captured? The signals are different. If you look at the signal part here, the signal is different. If you concentrate on this, the signal is different. If you are focusing on this, signal is again different. The FFT analysis on the source signal for reading unusually high delta activity is recorded. Unusually more high delta activities will be found there, will be observed, and far too slow during the cognitive processing. So when a mental task or some cognitive task is being done, the response will be very slow. More brain areas are actively involved during this cognitive task. And this is, the response in different montages, giving the, I'm sorry, giving these different peak values. User defined montage. Montage can be also declared and defined by the user. User wants to use electrodes in such a certain manner that is possible, that can be allowed to the user. Human BCI. We have studied a lot about human BCI where the brain controlled interface, brain computing interface or brain control interface, where we also know that they are mainly of three types: invasive, partially invasive, and non-invasive, where you have what are different types of task. One is a motor imagery task, and second is speller, which is actually your, what is called as a stimulator. So both of them are associated with, and both of them impact the brain area. Say when you watch something and then you react to it, so that also affects the brain. And when you perform something, you need that you have to move your hand by this much, so that also responds, that is also responded by the brain. So brain is always impacted either by motor imagery task or the visual speller, which is the stimulation kind of task. So in invasive manner, implanted directly into the gray matter of the brain during the neurosurgery, targeted repairing damaged site and providing new functionality to the person with paralysis. I think we have already had a detailed discussion on these. The invasive is a BCI which is recommended for the people who have severe disability like paralysis, and can produce the highest quality of signal because you are going deeper into the brain and covering a very smallest possible group of the neurons. So the quality of the signal will be definitely much better than the other two modalities. Direct brain implant was used to treat non-congenital acquired blindness, and William Dobell was one of the first scientists to come up with working brain interface to restore the sight, that is to work on the vision. This is the example of the picture highlighting the invasive, where you reach into the gray matter and insert your electrode with the help of surgery. So without surgery, you can't do this. Then comes partially invasive. In partially invasive, implanted inside the skull, but not at the top of scalp. Inside the skull and but rest outside the brain and produces better resolution than non-invasive, having a lower risk of forming a scar tissue in the brain than fully invasive BCI. Fully invasive BCI, there is a likelihood of risk related to a scar, wounds in your brain. So the example under partial, you know it very well, electrocardiography called as ECOG, which is used to indicate the electrical activities of brain taken from beneath the skull, just beneath the skull, not the skull. Electrodes are embedded in thin plastic pad and is placed above the cortex area beneath the dura mater. The first trialed in human in 2004 by Eric and Daniel Moran. In 2004, this kind of experiment was done. Then comes another method under partially invasive, which was not covered when we were discussing about the different examples of BCI, which is light reactive imaging. Therefore, in the beginning, I told you we are highlighting the fundamentals of biomedical signal processing. And that too, whatever we have studied, we are also extending the coverage of that topic or subtopic. So light reactive imaging BCI, it is still in the realm of theory. A lot of research is going on and it involves implanting a laser inside the scalp. You just send one laser inside the scalp. You know what is the role of laser? Laser is the trained on single neuron and neurons reflectance measured by separate sensor. So the laser, detects the activity of single neuron and it is measured with the help of separate sensor. When the neuron fires, I have told you the firing process. Whenever it emits certain high potential at certain peak, the phenomena is called as firing. So whenever the neuron fires, it excites the laser light pattern and wavelengths will change, and that is recorded back with the help of a separate sensor. What are the advantages of partial invasive? Better SNR, higher spatial ratio, and better frequency response, frequency range you get in the partially invasive. Then comes non-invasive, where you don't have any risk. Recorded signals are used to power muscle implants and restore partially movement. Signals are weak here, but the waves are still detectable. Whether signals are always weak in the case of uh, non-invasive, but still can be detected. But it's very difficult to determine the area of the brain or the neuron that created the signal, because the neuron or the group of neurons which has actually created the electrical signal is much deeper inside the brain. So therefore, it becomes very hard to determine which area of the brain from which particular specific area we are getting this particular signal, and therefore it gives the rough approximation. There are a number of non-invasive methods based on EEG, MEG, and MRI. EEG, you know it. ME is a magnetoencephalogram, and MRI is magnetic resonance imaging. So the method with non-invasive or all non-invasive methods, the advantages are ease of use is there. It's always portable and low setup cost. There is no specific setup cost involved in the non-invasive. EEG is most studied potential non-invasive interface with fine temporal resolution. As far as the temporal resolution of EEG is concerned, it is very good. So is very popular among all the research work. So if you talk about the history of EEG in mid 90s, Niels, University of Tübingen in Germany trained severely paralyzed people to computer cursor. There was an experiment where number of serially paralyzed people were asked to experiment, and they were to self-regulate the slow cortical components in their EEG. EEG signal was used as a binary signal to control the computer cursor. 10 patients were able to move computer cursors by controlling their brain waves, but this method was slow and it required at least an hour to write 100 characters, because the person was asked to use the cursor and write the character. In 2000s, Jessica Baylis, University of Rochester, they demonstrated the work, including a number of volunteers. The volunteers were asked to wear a virtual reality helmets that could control elements in virtual world using their P300 EEG recording. This is one such example, including turning light on and off. So they were asked to turn on and turn off the light, using the stimulator and P300 BCI. So magnetoencephalography, which is called as MEG, is used for mapping brain activities by recording magnetic fields produced by electrical currents occurring naturally in the brain. Clear? So magnetic fields are produced. Recording is not for electrical activity. Recording is done for magnetic activity. Activities are all brain activities, but recording magnetic fields which are produced by electrical currents occurring naturally in the brain. By using arrays of squids, superconducting quantum interference devices, this is possible. This MEG is used in localizing the regions affected by pathology before surgical removal and determining the function of various parts of the brain. Like sources of magnetic field will be there. Orientation of neurons are there because whenever there is a magnetic field, there will be north pole and south pole. So some of the neurons will be oriented to some pole and other neurons will be oriented to other. So active neurons are here, like scale, skull, or the different areas where neurons are oriented to certain direction. And then you have a device. With the help of that device, you are detecting the MEG, and then the field pattern you look at here, clearly see the poles, north and south kind of poles you can have. And then you can use certain model, classify the different areas, and you can determine whether the person is having epileptic or some other kind of seizures or some other problems you can analyze. Coming to MRI, used in radiology to visualize detailed internal structures of brain. So you might have heard when there is a treatment on a brain tumor and other tissues related difficulty, the doctors always recommend go for functional MRI. Rather, it's a type of MRI scan that measures the hemodynamic response, change in blood flow related to neural activity in the brain or spinal cord. I have already explained about what is relation between blood flow and the neuronal activity. So whenever there is a neuronal activity, that also affects the blood which is flowing in the groups of neurons. That is recorded by MRI, which is used to measure the hemodynamic response of the brain. Recent research in ATR, Advanced Telecommunication Research, Kyoto, Japan, on the functional MRI indicates the scientists to reconstruct images directly from the brain and display them on the computer. And this is how you can get at different points the different impact created in the MRI signal. EEG, let's discuss some more description about EEG, why the name is EEG, electroencephalograph. So actually here electro is for electric, and then CEP, cephalo means for brain, and then gram, you know that picture. So therefore it is called as encephalogram. Encephalogram is recorded with the help of electrodes. No need to repeat. We have covered electrodes on the scalp. They are used to measure electrical activities generated by thousands of synchronized neurons. Those neurons which are in sync and they are in group, that response is recorded by the EEG signal. Direct non-invasive measure of neuronal activity, really having good temporal resolution, and sampling rate of the EEG signal is generally in the range of 1024 to 4096 hertz with modern equipment systems with which we record the MEG signal. The physics of EEG, electrical potential generated by neurons, can be modeled with dipoles. This is giving you the physics, internal physics of EEG. Electrical potentials are there which are generated by the neurons. They can be modeled with dipoles. Dipole is a separation of electrical charges quantified by the dipole moment mu. There is a magnetic dipole, but there is electrical dipole also. You know it very well that when there are, a positive and negative charge carrier, they are separated by a certain distance. It forms a dipole. So the electrical potentials are generated by the neurons that can be modeled with dipoles based on the polarity. All right. So the electric current flows from negative pole to the positive pole of the dipole, and that happens in neurons all the time. And that is called as action potential, and that is actually the potential difference that is coming from the some part of the brain. Primary versus secondary current. So there is another factor, primary versus secondary current. First is related to the dipole. Second is related to electrical current, action potential. Third is related to primary and secondary current. Single current is very small to measure. So what happens that you have a group current contributed by all the neurons. That is only the way in which you can measure it. So this is you look at here, there's nothing but the dipole, positive and negative, and you all know that these are the orientations and the directions are all going from positive to negative. So secondary current is this much, and primary current which is flowing between the main path here is the what I can say the synaptic axon. This is actually a neuron. Look at carefully. So this is primary current, and all other currents which are flowing, considering that these are the two ends of the poles, are all called as secondary currents. So this is some more description and physics about physical phenomenon about the EEG signal. Then comes one more term in this study called as neuroanatomy. Neurocortex consists of 6 distinct layers, and distinct neuronal organization and connections across the layers are there, and giant pyramidal cells projecting from layer 5 are lined perpendicular to the surface. If you look at here, all the 5 layers have been described here. Activity in the cortex layer 2, and then this is 1, 2, 3, 5 here, and then this is again the description of 5 and 6. So axons are from there, and these are also axons. These are all axons, and activity in cortex 5 is recorded here. And this is your cortex, which is there, axons and dendrites, various layers. This is a white matter, which can be found here. You can see one neuron here, and these are the nerves which connect different neurons. And this is a pyramidal cell to other parts of the brain are connected with that. One more term that comes in the analysis of biomedical signal is pre-synaptic potential or post-synaptic potential. Presynaptic potentials are of small duration and biphasic. Whether there will be two phases you look at here, at least positive and negative side. So there will be two phases. So it's called as postsynaptic potential. Presynaptic, I'm sorry, this one. And whereas in postsynaptic, look at the direction, it's only one. It's not separating positive and negative. So postsynaptic is longer and monophasic. There is no phase shift. But here the phase is there. This side is different and this side is different. All right, so this example, which is of 2 millisecond duration, is of presynaptic, and the other one example was of the next one, which is the 10 millisecond, is the gap there. So what do we measure? From the EEG, how do we measure? What is the physics behind it? Summed up dipoles generated by synchronized postsynaptic potentials or tens of thousands pyramidal neurons in layer 5 plus noise is measured. So there are thousands of neurons that make pyramidal neuron. There you get one synaptic potential, and of course some noise can be added there. So this is one diagram where you can understand intracellular local field potential, and this is local EEG. So when you are going inside, then the next one is very simple amplifier. And then this is MEG pickup coil, EEG electrode and scalp. This is skull. Here you get the semi non-invasive. Then Dura master is here, primary intracellular current MEG flows here, and secondary, all these blue colors are secondary, and in the center, there is a primary color. And I think same kind of description has been given in the second one, effect on biopotential reading, and the axial dendrite, apical dendrites, where these are dendrites where the different types of charges you can see constituting the dipoles there. You get this type of current as a primary current or secondary current. Right, so electrodes placed on scalp, we generally use 10-20 system, international system, where between the two electrodes, somewhere is 10% of the total span and somewhere it is 30%, 20%. It uses differential amplifiers to produce each channel. You can see here that the amplifier which is being used here is a differential amplifier. Why do we use differential amplifier? Because we get information from two different channels. So there are different ways in which electrodes are connected to the amplifiers, and the ways in which we connect the electrodes, we have already studied in previous lecture. We call them montages. So this is one typical diagram of single channel EEG, where there is one channel from where the signal is coming, and then we can pass through either HPF or a bioamplifier, biosignal amplifier, and then if requires, we can pass through low pass filter, then analog to digital converter will pass through DSP, digital signal processor, and then with the help of certain graphical user interface GUI, we can see the result of what we are interested, what we are intended to, observe in the brain signal. So this first one is for multi-channel, and second is for single channel where you have one reference and only one electrode. So it also records and passes through the differential amplifier. It goes to HPF, amplifier and LPF, ADC, DSP and GUI, and this is how we do the analysis. So reference types, here there was a term called as a reference types. So what are the reference types? Common reference derivation. The reference node, you mean reference, what I can say, electrode. So reference derivation, reference electrode is subtracted from the scalp electrode, and same reference electrode is used for every amplifier. All right. So in the first case, you can use the same reference for both the points. I think I have already discussed in the previous lecture. Remember, FP1 plus say reference is FR, and this will be A2 plus FR. So what will happen when you subtract these two? The reference voltage is cancelled, but reference voltage is added with both the electrodes, considered with both the electrodes. Then comes average reference derivation. So here what happens that with every electrode there will be average reference electrode potential. So how do we get the final potential difference amplified by the amplifier? That electrode potential minus the average potential. So it's summed, averaged and passed through high value resistor, and the resulting signal is used as a reference voltage. Then comes bipolar derivation, where very simple, you just simply take, there's no use of using either to add a reference or to add a average reference. Here we simply subtract between the two electrodes, two nodes, and just get the difference as the amplified version. So these are the different reference types: average reference, and the reference electrode, average reference electrode, and third one is bipolar derivation, where there is in fact no electrode is there. So talking about the EEG spectrum plays important role in order to assess and study the different types of variations in the brain signals in different psychological and physiological and mental and neurocognitive phases of a human brain. So what are the waves? I need not repeat. We know that there are beta, alpha, gamma, theta, delta, where you see that delta has the minimum frequency, and all these things I've already explained, like beta symmetrical distribution from both the side. You find both the sides symmetrical distribution, and dominant whenever there is alertness, anxiousness, and eyes are open. We have already covered this part. Alpha in posterior region, it is found higher amplitude on dominant hemisphere, and that appears with relaxing and closed eyes. And then theta slow activity, delta lowest frequency and highest amplitude appears in stages 3 and 4 of sleep. This is very interesting. So when delta is there, we mentioned that delta is present in during sleep or meditation. So there are different stages of sleep. Every one of us undergoes those stages of the sleep. So especially the delta is observed during the third stage and fourth stage of the sleep. What are those stages? Probably included in the next slide. Look at here carefully. This is one diagram that describes stage 1 when you feel drowsiness, first 5 to 10 minutes. Then stage 2, you go for light sleep, last 20 minutes. It remains for 20 minutes, the light sleep. Then another 10 to 20 minutes, you'll have moderate sleep. And then next 30 minutes, you'll have deep sleep. So the moment you go to deep sleep, actually it takes at least 1 hour, 20 plus 20 plus 10, almost, 45 to 50 minutes it takes to reach to the deep sleep. So where are the stages 3 and 4? Either you are having moderate sleep or deep sleep, then actually delta is observed. Because when you are still in light sleep, your brain is working and thinking, engaged in other activity. You are thinking about something, some place or some person or some work, and therefore you are still not getting into the deep sleep or moderate sleep. And what is the last stage? And that longs for it's not written, rapid eye movement. Rapid eye movement, also called as REM. This picture is indicating all these. When you are awake, your brain wave will be like this. When you are in stage 1, look at the drowsiness, It will come like this, trying to settle down. Look at here. When you are in stage two, I mean, stage two is a light sleep. So it's again, further going down to settle down. And when you are in stage three and four, this is the pattern which we call as a theta, I'm sorry, delta. And when you are in rapid eye movement, this becomes almost less. So in the rapid eye movement, it actually helps you to revitalize your memory and intense dreams occur. So especially when you are in REM stage, sometimes you can dream of because you go, and actually that is the stage after reaching to deep sleep and then going to REM, rapid eye movement. Remember, you rapid eye doesn't mean that you again start blinking your eyes and you start moving your eyes rapidly. No, it means you are recalling something, you are going to revitalize yourself, and then sometime because of that your eye goes very far and you have some dream. So this is what is called as a rapid eye movement. So this is another interpretation of all the cycles of sleep. Stage one, light transitional sleep, drowsiness, and there are different ways in which interpretation is done. Stage two, more stable sleep. Deep three, growth hormone is released. Remember, there are one more interesting thing, and why do we need the quality sleep? Because look at here, in stage one, drowsiness and deep begins. Stage two, chemicals, more stable sleep is there, stage two, but chemicals block in senses, making it difficult to be woken. So there is some chemical which is emitted, and then that blocks the senses that actually make us awaken. So some chemical will be there, and when you go to deep sleep, growth of hormone is released, and that always nurtures your brain, and then therefore you get revitalized. Remember, everything is therefore needed. So when you go for a medium sleep, some chemical will block the senses, because until your all the senses are working, you will be always not taking the rest, and therefore you need a rest. So how do you get the rest? When you go for deep sleep, there some hormones are released, and then you get those hormones help you to get revitalized. This is a one kind of application where you can capture the EEG signal, and EEG can indicate typical brain waves during sleep, wakefulness. Look at here, this is the theta wave, and 4 to 7 hertz. This is during stage one, stage two, stage three. So in stage three, clear cut difference can be seen. K complex is one of the the what I can say component of your sleep during third and fourth stage, used as a metric to indicate the amount of or the quality of the sleep, like that. So talking about MEG signal now, some more description, and then after 5 minutes we'll have a break. MEG signal is actually provides, it gives direct external recording of magnetic fields which are created by electrical currents in the cortex area. Look at the diagram, you have to completely insert your head inside the MRI or kind of a magnetic field recorder machine, where, the electrical currents in your cortex area will be creating some magnetic field, and that will be recorded by this machine. So why the arrangement is always like this, even if you capture MRI, because magnetic field will be then interfered by the surrounding, and therefore you have to put your head completely inside this device. So neuronal activity or neural correlates with the cognitive or perceptual processes, either cognitive or perceptual, where you are trying to perceive something and localize affected regions before surgery, and also determine regional and network functionalities are reported. So this is how the it works, has been indicated that B field, this is nothing but a magnetic field diagram, laser beam is there, field coils are there. So this is how this is the arrangement of the device where you put inside, and then with the help of these cables which are there inside this device, you get the magnetic field patterns recorded in the form of signal. So that is what is the MEG signal. And you can see very clear cut difference between MEG and EEG here. In EEG, you see that plus minus potential charges are there, whereas in MEG, you have, this polarized magnetic fields are there between North Pole and South Pole. So EEG and MEG both, they are used to measure the neuronal activities, but EEG records synchronized electrical activities. I have told you, synchronized means all the neurons which are there in the group, they are synchronized and their combined activity is recorded of the large group of neurons, whereas MEG detects tiny changes in the magnetic fields. So whenever the electric current is flowing there in the synchronized group or the group of neurons, so how much change is coming in the magnetic field, that is recorded and seen in the MEG. Dipolar current flow through generates a magnetic field, and 10,000 to 50K active neurons are required to detect a signal. If below that number of neurons is there, you may not be able to observe the strength of the MEG signal. So you need to have a bigger group where the enough amount of current will be generated that will be causing the magnetic field to be created so that it can be detected. So that is there. So the scale and application, MEG signal is tiny. Interference from electrical equipment is always a problem here. It is affected by the traffic, the earth, participants' heartbeat, because that will also affect the pattern. It requires magnetically protected rooms and super sensitive magnetometers. Excellent spatial resolution is reported in MEG. It is good for functional mapping of specific cortex in a particular area during the behavioral, cognitive, and perceptive tasks. When you are interested to assess a behavior of a person, mood of a person, emotion of a person, and cognitive ability and perception capability of a person, then this is a good modality. And surgical planning in patient with brain tumors or intractable epilepsy, in such case also that planning can be done. Research into whole brain network connectivity can be made using MEG, and the temporal resolution is in milliseconds. So one diagram is there when you have functional MRI, stationary MEG, and other one is non-stationary MEG. So this is one comparison, and after comparison, we'll stop for a break. So this is a comparison for between the two modalities, EEG and MEG. Look at the EEG signal amplitude, 10 millivolt. You calculate the amplitude in magnetic case in Tesla. So it is 10 FT measurement. Secondary current is measured in EEG, whereas MEG you measure the primary current between the pole. But what are the secondary effect? That is actually your EEG. Distortion by skull or scalp is always there in the signal quality, and the little effect by skull and scalp in MEG, but external effect will always create the disturbance. The temporal resolution in both the cases is almost 1 millisecond. Spatial resolution is almost 1 centimeter in EEG, but lesser than MEG because it goes even deeper to the pole level. Outside the secondary current you are recording. So the spatial resolution is better in case of MEG. Experimental flexibility is so much in EEG, you can make it portable, but here you can't make it portable, and subject has to go and subject remains to be very stationary. And the dipole orientation, you have seen the dipole orientations in EEG are temporal and radial, tangential and radial, tangential components and radial, and here tangential is much better than the radial. There is one more diagram that gives the comparison. If you remember, this was already described in one of the previous lecture, comparing all the modalities in terms of temporal resolution and spatial resolution. You look at temporal resolution, see here, it is very good here in MEG. This is somewhere between 10 to the power minus 1 to 10 second. For MRI, look at PET. It takes 100 to 1000 second. MRI also takes around 100 second. And then EEG, it takes, some millisecond and then get this. So when you see the invasiveness is this side low, this side high, you are getting low invasiveness towards EEG. And this is in terms of spatial resolution. So three things have been included in this comparative chart or a diagram: invasiveness, spatial resolution, and temporal resolution. So our interpretation. Let us continue our previous part with the manual interpretation. If we interpret EEG signal and other biomedical signal manually, what happens? So you see here that a signal is captured with the help of one machine and you get the EEG signal. And then what will happen? The technician will administer a 30-minute recording session, and The EEG specialist, some person will be there who will be probably neurologist, will interpret the EEG signal. If you want to do the manual interpretation, will generate some report, EEG report, and will be further suggesting further diagnosis. If he finds that some abnormalities are there, actually you might have seen even if you go to doctor related to cardiac problem, he will capture the ECG, electrocardiogram. And in the electrocardiogram, he'll try to find some abnormality, wherever the pulse rate is looking, deviated from normal, then he will think that some difficulty is there in the ECG recording and will be either recommending for a further test or some diagnosis. So this is we generally do in manual process. But here what is the difficult? Number one, we do not have computer aided diagnosis method where computerized automated analysis. And when we say computerized automated analysis means it involves signal processing, correcting the noise, making your signal appropriate, then removing the noise and artifact, extracting the features, then appropriate, use of classifier. After that, then we will conclude actually that whether it is a normal or abnormal. If it is abnormal, how much abnormality is there? So you can understand that why in all modern diagnostic centers, in all modern hospitals, you will find the different equipments, a lot of computers, computer-based analysis, where the role of the doctors will be on the deeper analysis of the signal rather than the manual inspection of the signal. So manual interpretation, of course, it has a value, it has a significance, but the efficacy of the manual interpretation and augmentation of performance is improved to significant extent when we go for computer aided diagnosis method. So the common disorders which are diagnosed with EEG signal are, we know epilepsy. I think in previous lectures I have highlighted about this. Epilepsy is a neurological disorder indicated by sudden recurrent episodes of sensory disturbance. Sudden recurrent episode comes with a person suffering with epilepsy, loss of consciousness or convulsion. So sometimes we call seizures are there, you know, and suddenly there is a loss of consciousness. So it can be also studied with the help of EEG. Stroke, sudden disabling attack or loss of consciousness caused by an interruption in the flow of blood to the brain. So what happens? Suddenly the brain flow stops, which goes to the brain, and that is the brain stroke. So is there any possibility of brain flow stoppage, the obstruction and all that? That is very well studied with the help of EEG modality. The other one, the area, the disorder called as posterior reversible encephalopathy syndrome called as PRESS. Posterior reversible encephalopathy syndrome. What is this? It is characterized by headache, confusion, seizures, visual loss, and that may occur due to a number of factors, predominantly malignant hypertension, because there are two types of tensions. Hypertension, one is ignorable, one you can avoid. It may be because of huge workload, work task you are having some particular day or time, but malignant is not manageable. It's very difficult, and if you ignore malignant hypertension without any medication and treatment, that will further cause this kind of problem. Then eclampsia and other medical treatments may also cause this one. Other disorder called as middle cerebral artery, MCA infarct, obstruction of one of the three major paired arteries that supply blood to the cerebrum. So if there is any obstruction to the blood flow to the arteries, that causes this difficulty. Alzheimer's disease, and is a very common disorder related to the brain disorder and then sleep disorder, which is also called as narcolepsy. To determine the overall electrical activities of the brain in order to study all these disorders, which one we have to use, which one to be used is evaluated So, in even trauma, drug intoxication, brain damage, the electrical activities are evaluated. To monitor blood flow in the brain during surgical procedures, we can also use the EEG signal. And in all these disorders and disability studies, pre-processing, you know that in signal classification, the first stage is pre-processing, artifact removal, independent component analysis. If you have seen that video, there also this method was highlighted. Actually, it is considered as a very popular method for denoising the signal and removing some artifact or some unwanted signal. Deep learning systems typically use a dimensionality reduction technique because when we work with EEG signal, you know that huge dimension is involved, lot of channels and lot of signal features from each channel. So such as principal component analysis that helps to choose the important and meaningful components of the signal, and to pre-process means PCA or similar method when we apply to the pre-processed features and manage the complexity so that we can reduce the complexity of the the feature set. SVM like support vector machine, multi-layer perceptron models, and so on. These are used as a classifiers. And so the major stages when you actually want to classify your EEG data includes artifact removal, then we extract the features and then we classify. So we can use different types of methods, supervised method, unsupervised method to classify. So in the unsupervised, in supervised method, you know that your data is labeled, you have a target where to reach. So with a minimum, you have some what is called as a convergence condition, and you have to follow that convergence condition. There will be some minimum error rate. With that, you will be aiming it to reaching to the target. But in the unsupervised, it doesn't happen. You have to keep on working until and unless it converges and there is no further classification possible. So you begin with the test and you have already trained the model. You have trained model and you have feature set. So during the test, you again repeat, extract the features and use the suitable classifier. So in unsupervised, we can extract the useful features from unlabeled data. You know about unsupervised. Any unsupervised method, the data is unlabeled. It can remove redundant information from the input and preserve the unique aspects of the input. So in order to, let us classify the data in such a manner that reducing the redundant information, let's not repeat, and let us preserve the original information, which is very much needed, isn't it? So that we do. So ML, which is used to train the system, it automatically adjusts the weights of the different layers of the network such that it can replicate the input data at the output level. Of course, by reducing the redundancy and preserving the inherent content. We can extract the relevant features from the test data and can use classifiers to verify the validity of the features. What are the challenges in the feature extraction in the process? Sample time series data contains noise and the sample data is highly dimensioned. Both you have understood very well. It always has some unwanted signal and the dimensionality is a main problem. Sometimes data itself does not contain enough information so that you can actually get the features and make any relevant classification. So it does not contain some useful enough information to support the discovery of new knowledge, discovery of something new, and you can model a process that can help in classifying the data because your data is unlabeled. Critical characteristic of time series data. Remember, we are talking about time series data. Why? Because EEG, what we receive is a time series data. So particularly from stochastic or chaotic systems, is that an identical input can provide different outputs at different times because you don't know, it's not deterministic. So therefore it may produce different outputs at different times. So that uncertainty is there, which is a major challenge in the analysis of EEG signal. Developing domain specific techniques for modeling such complex time series data. So when you develop such domain specific, for one domain, for one specific application, you develop different model. So the process will be then time consuming and will require the subject matter expertise when you are segregating your entire features into different domain specific usages. So autoencoder, where you use encoder and decoder, this is an unsupervised deep learning technique called as autoencoder, and use deep learning technique. Encoder and decoders are there. What is the role of encoder? It preserves the essential features from the input data. The role of encoder is to encode the actual one, the meaningful, so it preserves the meaningful. Decoder restores the input data using the extracted features. So look at here in the input layer, the data is input. In encoder, the encoder will keep on preserving the input features, and the number of layers between the input and output layers may be multiple, but at the end of every layer, the encoder will keep on working and will keep on preserving the appropriate features corresponding to meaningful information. And finally, once it is decoded, you will get the data which is actually restoring the input data, and that too after reducing the redundancy and removing the repetition. So to reduce the difference between input and output as much as possible, we use this auto encoder. And the features can be used for classifying the data with supervised classifier also. Once you get the minimum feature set, feature vectors, you can also use then supervised classifier. Its performance of this method can be improved using advanced deep learning layers like LSTM and CNN algorithms can be used, because in the CNN and LSTM that capability is there, reducing the feature vector, and then by pooling and other mechanisms, you can keep on reducing the redundant data and keep on reducing the amount of data. So the signal processing, say in this particular example, you have a signal. When you extract a number of features, what do we mean? Say here in a time domain of, this is the time domain signal. So in time domain, what are the features which we can extract? I have mentioned that that you have a different types of features which are extracted with the help of signal processing. One is the time domain, second is time frequency, and third is your frequency domain purely. So in time domain, you can note the rising time, you can note the falling time. Rise time and fall time are very important, parameters in signal processing. And there are different ways in which you calculate the rise time. Sometimes it is calculated from 10% to 90%, some cases 0 to 100%, depending on what type of signal it is. Duration of spike, what is the duration of the spike where the spike has occurred? And then maximum peak to peak, what is the amplitude and what is the peak frequency, and so on. So these are actually, what I can say, the time domain values only, time domain frequency only. Now in the next part, as I mentioned, I'm sorry, next part, as I mentioned, don't worry, I have given you enough fundamentals of biomedical signal processing, EEG signal processing. What are the different methods which are used to pre-process? What different types of methods which are used to extract the features? What different types of classifiers used to classify those features? So we have talked about signal processing. In the next part, in the next 50 minutes, we are going to talk about only the application areas. What are the different applications, of the EEG signal processing, which we have not touched in the previous lectures, and how the process and the signal processing helps in order to deal with each one of these major, problems related to disorder and disability. Because I'm not going to cover the application related to robotics and BCI, because we have already done the applications related to robotics and BCI separately in our previous lectures. So mental disorder. Let's begin with mental disorder. So mental disorders or illnesses are actually the conditions that affect your thinking capability. You may lose your thinking ability. You may lose your feeling capability. Your mood can change. You may have abrupt mood conditions, and behavior may also change. And these are all covered under mental disorder or some illness. It also includes occasional or long lasting chronic and can affect your ability to relate to others and function each day. So your each day activity will be severely affected by the mental disorder problem. So a lot of mental disorders are there. Some of them have been listed here. Anxiety disorder, including panic, some person can get immediately panic, can make a panic, even in a normal situation. It's not very much abnormal. Obsessive compulsive disorder, popularly, known as OCD, obsessive compulsive disorder. Phobias are there, manias are there. I'll talk about phobias, manias very, very quickly in the coming slides. Depression, bipolar disorder, and then mood related disorder, eating disorder. Many people have eating disorder, because of disorder, people have different, eating habits. Post traumatic stress, some trauma has been there in his or her life. So the post traumatic impact is creating the stress in person's life. Then psychotic disorder, including schizophrenia. Schizophrenia is very, important name for psychotic disorder related to psychological problem. So this is one example where again, I'm starting to talk about mental disorder and disability and how do we use the EEG signal processing. Therefore, this is a diagram that tells us that we extract the signals, we choose the exact or we extract the suitable band, and then what do? We can calculate all of these or any one of these. We can make feature vector. You know that what is the meaning of feature vector? Feature vector consists of a number of features like see in trial wise when you are doing training, theta 1, theta e, or any phase or other related parameters. So there are feature vectors and there are RGB image. Maybe you can have some maps, some color maps or color impressions or RGB maps could be created, could be saved, and they are all these coming from the signal. And then we use different classifiers, and then convolution network and others for training and testing. So this is not a new diagram. So mental disorders are not caused by the character flaws. Steps to getting diagnosis. When we are interested to diagnose the mental disorder, what are the general stages? Medical history, a physical exam and possibly lab test so that medical conditions could be causing your symptoms and psychological evaluation that may answer about your thinking. You may be subjected to some psychological test where a psychiatrist or psychological experts would be questioning you, would be asking few questions, and depending on a response, assessment will be done that what is your psychological status based on your thinking, ability to think, ability to feel, ability to behave like this. And as far as the treatment is concerned, the treatment of mental disorder depends on what is the kind of mental disorder you are suffering with and how much seriousness, what is the stage of mental disorder, whether it is in early stage or in medium stage or it has become in the severe stage. Some people also need social support and education on managing their condition. If you remember, I was telling you the robots, social robots, I was telling you, if you remember, social robots are used to encourage the people, especially those who are, highly discouraged and demotivated because of long time, chronic mental disorder and disability. And there you need to encourage the people. You need to create awareness. You need to motivate them so that you can manage their problems, in such conditions, social support, education, proper awareness, and motivation are also provided in the process of treatment. Remember, this is not being done only by the psychologist or those who are counselors, but this is also being done by the BCI based robotics control and other BCI rehabilitative signals. That is a kind of, feedback signal we have talked about in our previous lectures. So in a few cases, patient may need more intensive treatment when it has reached to a severe stage, a very, the problem is very severe. There a lot of intensive treatment is needed. In terms of treatment in hospital, you'll get counseling. Generally, in the treatment of mental disorder, even though machines, equipments are there, diagnosis is taking place, is underway, but there will be counselors. We'll have a number of group discussions to involve the people, to express his feeling, to express his thought process so that he can come out, and activities with mental health professionals and other patients. So that will, revitalize his, his lost, senses that cause the brain disorder or the mental disorder. So what is psychological mental disorders or psychiatric disorders? are the patterns of behavioral or psychological symptoms. There is a specific symptom or a change in a behavior. There is abrupt change in the behavior or a pattern of symptom that is exhibited, demonstrated by a person that impact multiple areas of life. If you have some psychological disorder that impacts the life of a subject hugely and create distress for a person, he can't lead a proper and normal quality life where that causes a lot of distress to his life. So this is a generalized again diagram where you can based on the EEG signal acquisition, processing, extraction, and classification, you can even think of classifying different types of mood and emotions, and you can also suggest what kind of abnormality is there in the behavior of a person or a subject. These are some examples of mental disorders: depression, panic disorder, schizophrenia. I think these were all listed there. Post-traumatic bipolar. Dementia is a disorder. It is there in the old age where you keep on forgetting about the things. You lose your memory actually. That is called as dementia. And phobias and manias, you know it very well. What are the phobias and manias? So what are neurodevelopmental factors and neurodevelopmental task and the activities? The neurodevelopmental related disorder can be diagnosed during the infancy, during the childhood, neurodevelopmental, because the problem might be during the development of the child when even before his or her birth or adolescence and includes intellectual disability. So when you find that there is some lack of intellectual disability, you can assess with the help of a number of IQ testers which are available. And when you find that intellectual ability is less, the subject is exhibiting a disability in terms of intellectual, quality. So that kind of disorder that originates prior to the age 18 and is characterized by limitations in both intellectual functioning and adaptive behavior. You cannot adapt to, all the people in the society, and you can have less intellectual functioning. So it is generally identified or detected with the help of IQ test, and it is always considered, generally considered with the score under 70 that indicates the presence of that limitation. If it is more than that, considered as a neurodevelopmental, neurodevelopment is better. So adaptive behaviors involve, I was talking about adaptive behavior, practical everyday skills such as self-care, social interaction, and living skills. So the person will lack in practical life, everyday skills, whatever is needed for the self-care, for the social interaction with others, talking with others, behaving with others. Something will be there which will be abnormal in the behavior. This is, called as a cone, IQ cone. And this is one, test. There are a lot of IQ test available. This has been borrowed from www.freeiqtest.net. So it asks for some data actually. When you go to this website, it will ask for some data, and accordingly it will calculate the IQ test. And high profile people have been included, and their IQ was also calculated by using this, where some 161 was found for Einstein, Charles Darwin, it was 161. So you can see the values here, between 85 and 115, below average is 34.1%, and above average is 34.1%. Exceptionally gifted people, are 0.13%. Probably these are the guys. Super intelligence is exhibited by 2.1%. So 70% of people generally fall in this range. Those who exhibit from the value, this is value, remember, not the percentage. 85 to 115 is the value for most of the people, 70% of the people. So, but if it is very below, if it is the value is a 55, look at here, mentally inadequate, and low intelligence is there between 70 and 85. And even if it is below 70, there is a serious problem. So all such cases, the person can be, an attempt can be made in order to diagnose the persons having this much value of IQ in this range. Communication disorder, the person is not able to communicate, having some hearing disability, auditory disability, or speaking disability, that is communication disability. It impacts the ability to use, to understand, to detect the language, and to speak also, to hear, to speak, and to understand and to use something. So that is, the communication disorder. There are subtypes under communication disorder related to language disorder. You can stammer while talking if you have a problem related to the mental. Speech sound disorder, childhood onset fluency disorder, stuttering, this is called stuttering, and social, which is pragmatic communication disorder. You are not able to, mix with the society freely, and you are not able to communicate properly in a pragmatic way. Then there comes another kind of disorder called as autism and ADHD. ADHD already appeared. Autism disorder, autism spectrum disorder is characterized by persistent deficits in social interaction. If a person is not interacting with the society, and resist in interacting and communicating in multiple life areas, as well as restricted and repetitive patterns of the behavior, that comes as autism. That is also a mental disorder. Autism spectrum disorder is present during the early development period. This is generally developed. See, when you find a child who is not mixing with his or her friends and doesn't want to, you can say, you cannot say it's introvert, but is actually having some disorder. Because of that, he doesn't want to, interact with other people in the society. But you sometimes it is very difficult to, the assess whether the person is truly introvert or having some mental disorder. So the other one is attention deficit hyperactivity disorder. So if there is attention deficit, we were talking about one application, such application in the previous lecture, where a driver is driving and he loses the attention. So we can create, the attention with the help of alarm or alert, so that he gains his attention. And hyperactivity disorder, it is characterized by continuous pattern of hyperactivity. You find some person, always indulging, rather engaging in hyperactivities, and or inattention that interferes with functioning and presents itself in two or more settings, such as at home, work, school, social situations. Symptoms must have been present prior to the age of 12 as far as the ADHD is concerned, and have definitely negative impact on social, occupational, or academic functioning. That person suffering with ADHD will have negative impact in his or her life in terms of social involvement, occupational, as far as the academic performance. So this is one interesting, picture that gives you some idea about how does it impact in terms of signal processing for a person who is suffering with ADHD. This report was published, by the Cambridge University Press. Look at that. This is a time domain analysis where beta, alpha, theta, and delta are expressed, but ordinarily the beta, theta, alpha, the normal values are like this. So if you compare even your delta, look at the delta here, it's very different. See here. And if you compare theta, theta is also very different if you compare with the normal one. And the alpha has already been compared here. And if you find beta, beta is looking like this. So much difference is not observed, but in terms of, delta and theta, significant difference can be seen. And this I'm not going to repeat. You know that in problem solving, person exhibits no gamma. So I've just copied here. We have already included this picture in our previous lectures, highlighting about what kind of waveforms are generated by the brain, and each waveform indicates what kind of, ability or activity the person is involved with. Okay, so bipolar disorder is another disorder where the disorder is characterized by the abrupt change in the mood, as well as changes in activities and energy levels. I mean, you find a person who becomes extremely enthusiastic, and after some time you find that immediately he exhibits, less enthusiasm, less amount of energy, positivity. So it experiences between the changes in elevated moods, the depression, and are referred to as either mania or hypomania. These are called as bipolar disorder, mania as well as hypomania. So in such cases also the EEG signal processing is very much used in order to study it. Like this is an interesting diagram that tells you. Let's first read these statements. A mania is characterized by distinct period of elevated, expansive, and irritable mood, and that is accompanied by increased activity and energy. Sometimes it is marked by feelings of distraction. You are distracting by certain activity or a person, irritability, irritation you are getting, and excessive confidence you are having. People experiencing mania are also more prone to engage in activities that might have negative long-term consequences, such as gambling. So this kind of thing will actually make a person maniac to something. So say just to study this kind of study is used in terms of EEG signal processing. Like alpha wave, when you observe, when your eyes are closed and when you are in relaxed position, is like this. But when you are in different stages of mania, and other disorders, so irregular waves will be there in stage one. And then sleep spindles will be there when you are suffering with extreme mania, and the delta waves will start appearing rather than alpha. And in stage 4, it is purely becoming, mostly becoming delta. So the same signal in different stages of mania is becoming a different. So it's very difficult to predict, what kind of ability or what kind of activity the person is actually, indicating or exhibiting or practicing. So that is, the case where the mania is a problem which is related to mental disorder. Then there is another which is called as depression. Depressive disorder characterized by you feel that I'm depressed, mood is always sad mood, and along with the lack of interest in activity, person doesn't, focus on some activities and feel very sadness and doesn't find any interest in performing certain activities. And it may also involve other feelings like guilt because he's in depression. So guilt feeling will be there. Fatigueness will be obviously there because lack of energy, lack of interest, and will always feel irritated. During a depressive period, when there is a period when the person is having that disorder, people with bipolar disorder may lose interest in activities that they previously enjoyed. You'll find that a person who was enjoying that some work now losing the interest and experiencing the sleep difficulty, and even sometime, under extremely depression or depressive disorder condition, even thinking of, committing suicide. So that comes under this kind of disorder. Both maniac and depressive episodes, I mean, episodes are something, seizure is called as an episode. So that responses, maniac and depressive responses, can frighten for both person experiencing the symptoms as well as the family. So when you find some person having a maniac and depressive disorder, even the people who are around the person will always have, inappropriate experience. Friends and other loved ones will observe the behaviors and mood disorders. The treatment of such disorders include both medications, psychotherapy, where you say psychotherapy means rehabilitation, neuro, feedback kind of scheme will attempt to help the person who is undergoing with certain depressive disorder. That can help people with bipolar disorder successfully manage their symptoms. Anxiety is one more where it is characterized by excessive and persistent fear, worry, anxiety, and related behavioral disturbances. And a fear involves emotional response to threat. When you have some threat risk, so you have a feeling that some threat is there in your career or in any part of your life, you feel some fear whether the threat is, sometime it may be perceived also. Actually, the threat is not true, but you are in a different mood and you are scared, and you are unnecessarily worrying about. So that is anxiety, which involves anticipation that future threat may arise. So you're also thinking that, sometimes in a future some bad thing will happen and threat will may even become, what I can say, uh, more intense. So that causes the intensity. Generalized anxiety disorder, there is a name called as GAD, is marked by excessive worry about everyday events. And this is one, I think, picture that tells about the map. You see here in the map, there is a clear-cut map where there is no, it's a map of your brain actually, where there is no, impression, no activity. But when you compare this with the ADHD person, the person who is having ADHD, the person who is having depression, you see in the depression in a particular area, a lot of response is being created. Anxiety, it is further, increasing, and in migraine also you see that that thing is happening. So in all such cases, you can clearly see the change in the brain pattern, and this could be recorded and analyzed and could help the people who are diagnosing, who are involved in the diagnosis and treatment of the person. Of course, in the design of psychotherapy, in the design of rehabilitation techniques, also these responses could assist and try to motivate the person, because anxiety may be, as I told you, maybe due to the real threat, real problem, or sometime maybe due to the perceived threat. So if it is due to perceived threat, the person can definitely be motivated, counseling can be provided and can be corrected. So as I told you, if you remember about the previous lectures, we were talking about the feedback. The feedback may be auditory feedback, the visual feedback, or even the tactile feedback. So if it is an auditory feedback, the counselor can provide a particular counseling based on the response, where and what area and what kind of therapy the person is needed. And if required, some pictures can be, the person can be asked to see those pictures, and believing that those pictures, sudden impressions can, reverse the process which has caused unnecessary anxiety. So look at, this diagram I have already explained. There are different parts. Say this particular part called as a frontal area, where, manage negativity, explain and decode. So if the person is able to explain something and decide, that is determined by this area. The person who can manage the negativity is determined by this area, isn't it? The person who has who can hear voice tone and weight intent, and then the person who exhibits hostility for some other person, is the ability or the activity exhibited by this area. The person, say, notice errors, if you can find out and detect the error, if you can manage certain processes. So you can see that the different areas have different functionalities. I have already explained about a similar diagram in the previous lecture. You can just refer to the previous lectures which are available in the YouTube videos. So it involves an extreme fear, other phobias. What are different types of phobias, possible phobias? Maybe, an extreme fear of specific object or a situation in the environment. You are extremely scared about some object. So common is specific phobias that include fear of spiders, fear of some object, some insect, some animal, sorry, fear of heights, that height phobia, or fear of snakes. So these are some more natural events like thunder, lightning, tornadoes. You might have experienced, you might have found some people, when there is a thunder, they just get scared very much, and when there is a lightning, they'll get scared. So medical related phobias, medical procedure, you will find some person, highly scared of going for medical procedure, even with the, needle which is used to inject the medicine in the body. Some person you will find will be so much scared, and will always have phobia. And the dental procedure, the medical equipments, no, it's MRI machine, I can't insert my head inside the MRI machine. Such phobias are there. Animals, the phobias related with animals, dogs, snakes, bugs, situational. I mean, some persons are not very much comfortable with a small areas, maybe crowd or a driving or leaving home. If you have a phobia for separation, you are separating from your family, separating from your mother or a parent, that also is a kind of phobia. You can't manage it. When confronted by phobic object or a situation, people may experience nausea, trembling, rapid heart rate, and even a fear of dying in the case of phobia. Especially in the height or lightning, the person will think that, oh, I'm just going to die. So that much scary, the effect is there of different phobias. Okay, I'm sorry. All right, so these are some phobias: fear of being alone, fear of crowd, fear of leaving home, anxiety, feeling of detached from others, fear of public places are some examples. Then there is panic that also creates problem. Psychiatric disorder. Panic is a psychiatric disorder characterized by panic attacks. Experience a person will experience anxiety and preoccupation over the possibility of having another panic attack, and the people may begin to avoid situations and settings where attacks have occurred. The person will always avoid going to that place or doing certain tasks where he felt panic before or where they might occur in the future. That worry will always be there. This can create significant impairment in terms of the mental disability. Separation anxiety, I was talking about an excessive amount of fear or anxiety related to being separated. People are familiar with the idea of separation anxiety as it relates to young children's fear of being apart from their parents. The child knows that he has to, go separate from the parents, but older children and adults can experience as well. It happens sometime. The person suffering these symptoms may avoid moving away from home, going to school, getting married in order to remain in close proximity. So, having a anxiety based because of separating from family, separating from parent, all such anxieties might not be related to the brain disorder. Sometimes you are so much close, you are so much affectionate to someone, so you feel that when you go away from the person or from place, you're going from your comfort zone. So sometimes that gives you uncomfortability, discomfort. So that may also cause. Yeah, any question is there? Okay. Then there comes trauma. I am quickly completing all such areas where a lot of BCI, a lot of cognitive science, a lot of biomedical signal processing is applied, in fact. So those application areas I'm just telling you so that you can explore these areas, or you can at least, this is very important to know all these areas where the brain disorders and disabilities affect our life, not only for research, studying and getting the grades, but rather we also need to be very much aware, have to be knowledgeable about certain situations and stressful the activities that can actually create certain brain disorder and disability. So trauma or stressor related disorders involve exposure to successful or a traumatic event. Sometimes, a person becomes extremely successful. So feeling that also sometimes it gives the traumatic situation. Acute stress disorder, which is characterized by emergence of severe anxiety for up to 1 month period after exposure to traumatic event. So post-traumatic disorder is also there that makes some person going to the extremely, deeper in a stress situation. Such some examples like traumatic events include natural disaster, war, accidents, witnessing a death, and so on. Some person, attends a death. Some person has witnessed the accident, a major accident where the people were killed in a war. So such things will cause, the trauma, in a person's mind, person's brain, and that is a mental disorder. As a result, individual experiences disassociative symptoms. He'll say that, no, let me disassociate with these kind of situations where I don't want to undergo with the trauma, as a sense of altered reality, inability to remember important aspects of event, and vivid flashbacks as events were reoccurring. He will be going back, and thinking that, oh, things are again repeating, war is again going to happen, the person is dying, that much, that kind of thinking will again come in his brain. Adjustment disorder, very simple. It occurs as a response of sudden change such as divorce, there is a loss of job, end of close relationship, move, or some other loss or disappointment. So whenever there is sudden such thing happens, the person is not ready. So that definitely may cause some disorder. So you have to adjust to that. If you're not able to adjust, it will cause your brain disability. This type of psychological, what we are discussing are all mental and psychological disorders and disabilities we're talking about. The specific study on mind, behavior, psychological impact, application of cognitive science in studying the psychological impact, mind and behavior will be covered in the next and the last lecture on Monday. So, but of course, what we are discussing today are psychological and mental brain related disorders that affect both children as well as adults. Adjustment disorder is characterized by the symptoms. What are the symptoms by which we will know that the person is having adjustment disorder? Anxiety, irritability, depressed mood. The person is behaving very irritably, and he seems to be depressed. The mood is a very depressed, very low mood. Sadness is there, worry is there. You can clearly see the worry in his face. Anger is there, hopelessness. He believes that everything he sees in a pessimistic manner, and feelings of isolation. So these are all related to this. We all face such situations in our life, something or other we always face, when we change the place, when we lose the job, when we change the job, something like that. When we have to adjust, means we are leading normal life. So post-traumatic stress disorder can develop after an individual has experienced exposure to actual or threatened rape, serious injury, or sexual violence. Such kind of things will cause adjustment disorder. Symptoms of PTSD. What is PTSD? Post-traumatic stress disorder. It includes episodes of reliving or re-experiencing that event. You will say that, okay, you will think that again it will repeat and it will damage your life. Avoiding things that remind the individual about that event, feeling on an edge, and having negative thoughts. Dissociative amnesia. What is this? Temporary loss of memory as a result of dissociation. In many cases, this memory loss that lasts for small period of time or sometimes many years is a result of some psychological trauma. Some person undergoes with a psychological trauma for a long time. So there may be loss of memory for small time as well as for years together. Those who experience this disorder may remember some details and may try to reconnect, but is still not able to reconnect. So the events, but may have no recall other details around the circumscribed period of time. So they are able to recall something, some part of the event, but not completely, able to connect with other parts. So there is a memory loss called as a dissociative disorder. There is one more called as somatic symptom disorder. It involves preoccupation with physical symptoms that make it difficult to function normally. This preoccupation symptoms result in emotional distress and difficulty coping with daily life. Yeah, not even a simple, problem or challenge that comes in our life, we are not able to cope up with that. So it's kind of a symptom disorder. Somatic symptoms do not indicate that individuals are faking their physical pain, fatigue, or other symptoms. So you hope you can understand. Sometime, whether the person is uh, a person is having actual pain or not, person is actually demonstrating the true fatigue or not, is very difficult to find out if a person is having certain such kind of symptom disorder. Insomnia, you know it very well. This is related with a sleep disorder. We if we are unable to get enough sleep and to feel rested, this is accompanied by significant distress and impairment over the time. And hypersomnolence disorder is characterized by excessive sleepiness despite an adequate main sleep period. Even you get adequate sleep period, but you are not getting proper sleep. People with this condition fall asleep during the day at inappropriate time. Sometime in a school or in your workplace, you may suddenly get a sleep. And actually during the sleep period, you are not getting the sleep. Neurocognitive disorder. Major and mild neurocognitive disorders have primary feature of acquired cognitive decline, and more areas like memory. There is some declinement in the areas of your brain related to memory, attention, language, learning, and perception. All these areas you have found in some part of the brain. Somewhere in frontal, something is in the parietal, something is in the occipital. So cognitive disorders can be due to the medical conditions like a person is suffering with Alzheimer's disease, Parkinson's disease, PD, substance medication use, vascular disease, and so on. Like Parkinson's disease leads to, sacking the hands of the body, stiffness, difficult with walking, you are unable to walk and make balance and coordination, therefore you are unable to walk. So this is one such case where, this is the normal a person's, the brain. In an abnormal case, you'll have pain, depression, anxiety, behavioral changes in case of, Parkinson's disease. And that there will be distortions and constipation, other problems will be there in other parts of the body as well. And this part of the brain will be impacted in terms of signal processing when you have Parkinson's disease. Alzheimer's disease, we are talking again and again about this. This is very difficult to cure, irreversible generally. Progressive brain disorder that slowly destroys complete memory and thinking skills, eventually ability to carry out even the simple tasks. So person will not be able to even perform a simple task when the Alzheimer's disease is there. But of course, there are diagnosis and treatment procedures. And what is important in all computer-aided diagnosis, remember, when the disease is diagnosed, rather detected at early stage, you can always think of providing the care and treatment in the diagnosis. So this is, when you are awake and your signals from different areas, EEG. And I think this diagram is not very new. You get a EEG signal, raw signal in the training process, pre-process, feature extraction, and those feature sectors vectors are stored in the database. And in the train testing, what do? You do the same. You just classify that whether the feature set which is being captured of the raw signal is of a healthy person or the Alzheimer's person. And those data sets are already available in the data set. So you will just match and can tell the person is suffering with or person is indicating the symptoms of Alzheimer's disease. There comes the role of early detection of certain disorder. Schizophrenia, I mean, it appeared again and again, is a chronic psychiatric disorder that affects a person's thinking ability, feeling and behavior, complex long-term condition, which affects about 1% of people in the US and few other countries. So see the severity of schizophrenia is very common these days, as a person is losing ability in terms of thinking, feeling and behavior. What are the symptoms? Delusion, hallucinations, disorganized speech, all these are the symptoms of schizophrenia. Deluged beliefs that conflict with reality. What are hallucinations? Seeing or hearing things that are not actually, existing or appearing. Disorganized speech, words do not follow the rules of language and may be impossible to understand. So this is one example where you have the normal signal and the signal of schizophrenia person, and you look at that the difference which is studied, and then the symptoms are indicated. So computer-aided diagnosis, I'm telling you that computer-aided diagnosis is used for analysis of depression and disorder. So the complex and nonlinear, we know that brain itself is a complex. So our signals are also non-stationary, non-linear and complex systems. So very tedious to interpret visually and highlight, difficult to extract the significant features from these areas. So there are a number of methods which are used, linear and nonlinear, which are effective in identifying the changes in EEG signal so that we can detect the disorder. There are methods. Linear methods do not exhibit the complex dynamic variation because that's a linear. It will follow certain rule and it will simply produce the output in a proportionate manner. I mean, thus chaos theory and nonlinear dynamic methods are therefore popularly used as the nonlinear methods to extract the EEG signal features, which are used in the computer-aided diagnosis CAD-based system for the studying and diagnosis of depression. CAD system is simple to use and may be used by clinicians as a tool to confirm the diagnosis. So what is CAD? All modern medical facilities, diagnostic centers, hospitals, they all have CAD. Go to the eye specialist, go to the dental specialist, go to the cancer specialist. Everyone is having CAD based system. Now their diagnosis and the treatment has tremendously changed. They are so much depending on the machine, so much depending on the computer, isn't it? So this is one sample example of CAD based analysis. You see, you have EEG, I mean, auditory oddball task. I have already explained you what is oddball in my previous lecture. Number of subjects, number of electrodes, and P3B. I have talked about P3A and P3B. Then we use some certain pre-processing and then extract the features. You may have 16 time domain features, certain number of frequency domain features, certain number of features from electrode. So you can also choose certain number of electrodes, certain number of channels, and then channel grouping can also be done. You can select some electrodes from frontal, some particular electrodes from central area, and so on. So you can choose channel, you can choose features, you can select the features. I have already given examples in my second or third lecture when I talked about cognitive workload assessment. I have given the practical work which was done by PhD scholar based on how do we select the channel, how do we select the features, and how does it impact the work. And then you have a classifiers which are used whether the person is suffering with schizophrenia or some other disorder, or whether a person is a healthy person, can be done. So don't worry, I'll complete in time. As per the latest WHO report, the depression affects more than 350 million. Can you imagine? Of all ages, making it one of the most prevalent global diseases. This is estimated that a largest proportion of people who are suffering from depression are women aged between 18 and 45. Due to ignorance, untimely detection, we simply ignore that I don't have any problem. Misdiagnosis or improper medication, we sometimes, many people, will treat themselves, suffer from the disease worldwide. Untreated depression may lead to suicidal attempts as well. Global burden of depression disorder is increasing day by day. There is a need for comprehensive, coordinated response from all health and social sectors to encourage the patients and to subject them for the treatment, to encourage them. The identification of depression in the early stage is crucial to prevent it from reaching a severe and irreversible stage, so that it does not reach to the irreversible stage. Otherwise, there won't be any scope of improvement in the and the treatment. So this is one example where you see the normal left signal of the brain, and when it is in depression stage, see the lot of changes there. This diagram, I think it has already appeared for another case, Alzheimer's. This is classifying whether the person is having depression or the person is normal. The process is same. We train the model, we get the parameters stored in the database, and while testing, we find out whether the subject is exhibiting the property of characteristics of normal person or some abnormality. So then there are a lot of studies on dynamics, EEG dynamics, symmetry, asymmetry, resting the potential, how much variability is there. A lot of analyses are done. And the effects of drug is also there that accentuates the beta activity. The person who is addicted to drugs will have accentuation in the beta activity that will also cause the mental disorder. Medication for long time also augments the voltage or beta activity. Central nervous system stimulants such as cocaine, and other antidepressants may excite significant beta activity at low voltage. So persons who take unnecessarily medicines to reduce the stress in other levels, that will also cause the mental order disorder and disability. And then there are some methods which are linear methods I was telling you are used. They work on some amplitude and frequency and power features and work. There are nonlinear methods which are also used for the treatment. And what are the features? You can see bispectrum, power spectrum, phase entropy, wavelet energy, correlation dimension, fractal dimension. All I think I have already discussed in my previous lectures. Even today also I have highlighted to some extent. CAT system is very useful for diagnosis of disorder. It can it involves feature extraction, ranking, and classification. There are different nonlinear methods which are listed. Some of them are, like a fractal dimension, recurrence, quantification analysis. They are also used where you can do the feature ranking, and so that which feature is better, which feature is more useful, and so on. Highly ranked features are subjected to the classifiers. And of course, when we are talking about highly ranked features, we do not have to forget about highly ranked channels also. So we determine highly ranked channels and highly ranked features. If you remember in one of my previous lecture, I was talking about Prometheus method. That Prometheus method determines which method is a better ranked method that is giving you better classified output. So those things could be used in order to find out that which rank, which channel is better, which feature is better. Like you can see the difference between a normal person and the person who is having depression, even in terms of spectrum and even in terms of time domain signal. This is again a plot, a recurrence plot between a normal person and a depression person. This is given better in the next slide. Look at here, in terms of time domain, in terms of your that recurrence plot, which is an actually advanced technique for used as a nonlinear data analysis, where you see the visual, where you see the visualized square matrix, where the elements correspond to those times that which state of dynamic system occurs. So all those states where something wrong has happened, it creates certain impression. So you can clearly also find the difference in terms of recurrence plot as well. Rehabilitation, people with severe neurological impairments face challenges in supermotory functions. So I have told you already, I have described you that rehabilitation measures are used and they very well actually work as a feedback signal we provide. And there are ongoing research on non-invasive methods so that with the help of communication signal, feedback signal, rehabilitation can be provided. I mean, we have already discussed that how P300 BCI system helps the people who are elderly people. So there are approaches which are used to help severely paralyzed, locked in patients, communication using three different BCI modalities, slow cortical potentials, sensorimeter rhythms. We have talked about SMR, P300 potential as an operational mechanism. So I think there are 10 more slides. Let's not make hurry. I mean, Monday we are still have that day. I'm going to focus on psychological study. So we'll stop today here. This slide talking about rehabilitation. Rehabilitation has a tremendous potential, in the area of psychotherapy, neurofeedback signal, so that the equivalent analysis of what is the abnormal thing, what is the loss in the mental, framework, and how it could be recovered. Could it really be recovered? Attempt is made, and accordingly, based on the feature analysis classification, some equivalent feedback signal is generated, either auditory signal or a picture or a image or some tactile signal that is subjected to the person with the help of the equivalent signal, and attempt is made to rehabilitate and correct the disorder and disability to some extent so that person can lead happy life.